Right, hello. So we're going to quickly do folliculogenesis, which is nice and easy. So it's just the formation of the follicles. We've got three steps: a primordial follicle, a primary follicle, and a secondary follicle. So in this primary follicle here, what you can see, this little yellow thing here, is surrounded by a single layer of sort of like cuboidal low columnar cells. And then they're sat on this red line here, which is a basement membrane. And then around here is something called the theca. So let's just highlight these. The stromal cells comprise the theca, and they're just supportive fibroblasts and some collagenous tissue at this point. We have a BM, a basement membrane. Then we have these follicular cells, these flattened, slightly columnar cells. And then we have a primary oocyte. Now the primary oocyte is arrested at meiosis 1, the prophase stage. Okay, this is sort of like the whole ball game, if you will. This is the thing that's going to be developing into a secondary oocyte, and then will ovulate. Everything around it, so those follicular cells, the basement membrane, the stromal cells, everything around it is nourishing and protecting your little oocyte there. Now, every month, as the menstrual cycle comes around, FSH and LH levels fluctuate, and FSH in particular, follicle stimulating hormone, is stimulating the follicular cells to increase the nourishment and secretions of estrogen and other kinds of things to stimulate the primary oocyte to continue its development. So, if we move on the primary follicle, we can see a few things have changed. We've still got this primary oocyte, right? But now it's surrounded by a green casing. It's not actually green, but I've drawn it green here. That's called our zona pellucida. That's the protective layer from which the egg will have to hatch. Okay? Surrounded, those follicular cells that we saw previously now have proliferated quite massively and become granule cells. Now, the granule cells contain loads of granules because they're highly secretory. So they're doing a very similar role. They're just the same kind of thing as follicular cells, but they're slightly further developed. And instead of being a single layer around the primary oocyte, there's loads of them. So let's put some of these labels on. We've got our primary oocyte, as we did previously. We have a zona pellucida protecting it. We have a granular cell layer surrounding, again, nourishing, sat on the basement membrane, and then we have what's now called, from the stroma cells, further development, a theca folliculi. Now, what's the difference here? Well, when we go to a secondary follicle, we get further developmental changes to the oocyte, but we also get differentiation of this theca folliculi. You can still think of this as a protective layer of collagenous fibroblastic cells and the like. So here are the changes with secondary follicle development. So this is now what we call a Graphian follicle eponymously. This is a mature follicle that is ready to rupture. And what you see here is a few different layers. So we have an antrum, which is a fluid-filled space. So remember these granule cells here. What they're doing is secreting fluid into this space. And it's pushing your oocyte down and down and down. It's basically trying to get it to breach through the basement membrane and your theca. This happens to be the theca interna. There's also a theca externa. If it breaks through those two layers, and the inner layer of the theca interna, secretes loads of estrogen helping to power this, and then the theca externa is still stromal and protective. If FSH continues to be dumped onto this cell, causing estrogen to rise and all these different kinds of changes, then this will develop until the antrum grows so big, you see a blister on the surface of this follicle, and then, because it has only one place to go, the blister causes a bursting, boom, the secondary follicle ruptures, and the oocyte is released into the peritoneal cavity to be grabbed by the fimbriae and brought into the infundibulum of the oviduct, or uterine tube, or fallopian tube. And that's what the secondary follicle is doing there. Okay? This is sort of like a little schematic I've drawn there. So these little squiggly bits are the granule cells. We have an antrum here. Yeah. We have the theca layers here and the basement membrane onto which they're all sat. So we'll have a theca interna here and we'll have a theca externa here. This is our zona pellucida with our oocyte inside. Now what I want to draw your attention to is this little band around here. So a level of granulosa cells tightly adhere to the outer bit of the zona pellucida. So these will jump on here. When they tightly adhere, that is called the corona radiata, just like you have a corona radiata after the internal capsule in the brain. Okay? So you have a corona radiata, you have a zona pellucida, and then a secondary oocyte at this point. This oocyte is now secondary, which means that it has entered meiosis II and arrested at metaphase. Okay, so we have a secondary oocyte, we have a zona pellucida with its corona radiata intact, we have the bilaminar theca, we have the basement membrane, and we have that growing antrum, which is the reason we ovulate. Here's a scanning electron micrograph, so you can see what's going on here. So this is a follicle sat in the cortex of an ovary. And you have here, you have an oocyte. This lovely thing here, which you can tell, is the zona pellucida, and you have a corona radiata around the edge here. Here you can see an antrum forming. Here's a little blister. And here's what it looks like with histology as well. So a massive antrum. This is a very mature graphian. Here's your oocyte. Here's your zona pellucida. So you've got two little antra here. So you can see how in this finite space here, with the theca being supportive, in this finite space, it's going to grow and grow, this antrum, with the granule cell secreting until it pushes out our secondary oocyte. And there you go, that's folliculogenesis.